late division in a hypotrich ciliate. During the process of division, the parent cell, which is on the viewer's left, will undergo binary fission, forming what is virtually a pair of identical daughter cells. In this video, a transverse constriction has been formed in the parent cell, which is proximal, and many structures such as the nuclei, the lapel of the adoral zone of membranelles, vacuoles, and cilia have already been replicated and are visible in the developing daughter cell, which is distal. As this process continues, the symmetrical development of the two product cells proceeds rapidly over a span of several minutes. It should be noted that none of this seems to have hampered any routine activities, such as cell movement. Note that the actions of the two sets of cirri of the adoral zones are surprisingly well synchronized. Note that the crease or waist of the conjoined organisms is becoming progressively narrowed. The smaller globular organisms jumping, if you will, in the background are halteria. The two product cells appear as though they will ultimately be virtually identical if reproduction continues on its present course. The anterior end of the posterior forming organism is connected to the posterior end of the parent cell. Therefore, the last major organelles to be constructed will be the cirri of the posterior organism, particularly the collar. During this next period of the video, the organisms that are developing together will be more or less immobilized or stable, and this will provide an opportunity to verify all of the aforementioned observations. Note that both organisms have now developed visible contractile vacuoles in more or less the same ventral anatomical location. They too seem to have synchronized activity, both filling and emptying. Although at times the anterior vacuole seems to be somewhat in advance of the posterior one in the cycle. During this next period of the video, the organisms that are developing together will be more or less immobilized or stable, and this will provide an opportunity to verify all of the aforementioned observations. The process of completion of development and separation continued unabated for several minutes. The organisms were lost from view, and the actual separation is not visible in this video. Shortly afterwards, one of the two product cells became visible, and it is the subject of the next few slides. This is one of the two product cells under higher magnification. The two inset diagrams from Kahl illustrate the coarse, stylus-like anterior cirri, the cirri of the lapel, the finer posterior cirri and cilia, the cytopharynx, the nuclear lobes, and the contractile vacuole, all of which are quite comparably seen in the video, which is playing on a loop. Superimposition of the diagram from Kahl on the photomicrograph of the organism with gradual dissolution of the diagram provides a sense of security about the likelihood of the organism's identity. The illustration in this slide is taken from a collection of 320 drawings of hypotrich organisms in Kahl's text. It appears that the organism is Stylonychia and possibly Stylonychia muscorum. In summary, then, we have had an opportunity to see some of the events that occur in the late fission of a hypotrich ciliate.